Now that we're recording, we'll do that all over. That was actually moving on. Okay, my name is Rondy Malik. The title of this talk is Can Subsurface Plant Organs Attenuate Anthropogenic Activity and Stimulate Ecosystem Process? Okay, the burning of fossil fuel, deforestation, as well as land use management has led to the enrichment of atmospheric CO2. This can have ramifications at multiple levels of biological organization, including the ecosystem scale, such as net primary productivity, the organismal scale, such as photosynthesis, as well as the molecular scale, such as resource quality, particularly C to N ratios. And also, we could expect this to affect ecophysiological interactions, such as CO2 through the stomata and plant gas exchange can impact guard cell regulation, as well as stomatoconductance, as well as root respiration and dynamic root activity. Traditionally, we've thought of the role of roots as being a role in anchorage, absorption, and storage. And by anchorage, I mean gravitropism, which anchors the root to the soil, absorption, which allows nutrients and soil solution to be taken up at the root-soil interface, as well as storage, which allows carbohydrates to be stored in the roots. But also, roots can fuel soil organisms through phytochemical activity, as well as root turnover, detached root caps, mucilage, border cells, as well as small molecular weight compounds such as strigolactones, which allows the recruitment of mycorrhizal fungi, which can then colonize root cortical cells. So the flow of carbon below ground involves enriched atmospheric CO2, which is first captured from the atmosphere, and then which is converted into photosynthase, and eventually the carbon flows down into the roots, and the roots are able to exchange this with mycorrhizal fungi in return for nutrients such as nitrogen and phosphorus. So what's the big picture, you ask? Well, my research agenda is to address environmental change, anthropogenic activity, as well as microbes. I'm interested in the role of environmental change on microbes and whether or not microbes can respond and potentially mitigate environmental change. And two of the main umbrellas that my research fall under, I ask these two questions. Do microbes mitigate and do microbes respond? So today I'm going to tell you about two research stories or projects that are currently in review. The first one is accepted with revisions at plants, and the other one is in review at forest ecosystems. And in the first study, I turned to the study system of milkweed, atmospheric CO2, as well as mycorrhizal fungi. In the second story, I'm going to focus on differences in mycorrhizal types, such as the arbuscular mycorrhizal fungi, which colonizes inside the roots, as well as ectomycorrhizal fungi. And I'm going to focus on how decomposers respond to the soil conditions that is orchestrated by these two different mycorrhizal types. Okay, do soil microbes mitigate? First, to address whether or not soil microbes mitigate, I'm going to tell you about some environmental change that's been taking place over the past few decades. The past few decades, there's been a decline in monarch populations, as you can see here on this chart, that there's been a decline in monarch populations since, on this scale, we see since 1995. At around that same time, since 1995, we see that there's been an increase in atmospheric CO2 levels. Now, although we see these two patterns, correlation is not equal causation, but we have noticed that there is some environmental change happening, whether it's increase in atmospheric CO2, as well as the decline in monarch populations. The monarch populations may be due to decline due to their adaptive resource, which is milkweed. So it may be the case that CO2 may be influencing monarch's adaptive resource. And this can be due to the change in the resource quality. So this is much at more of a molecular scale, where you're probably getting an increase in C to N ratios, And perhaps this secondary metabolite, which the monarch larvae is able to usually take up from the plant and sequester in its tissue, perhaps when this dose of toxin increases in concentration, perhaps it becomes toxic to the monarch. Usually it's not, but that's just a hypothesis. And also these cardenolides, they're important because they have pharmaceutical implications. They're known as cardiac glycosides. Okay, as you recall, this is the flow of carbon, enriched CO2, converted to photosynthase, and then converted into secondary metabolites, such as cardenolides, which is stored in both the shoots as well as the roots. And the carbon could also be transferred to mycorrhizal fungi. And it is our hypothesis that mycorrhizal fungi could perhaps restore the plant quality. So by balancing nitrogen and phosphorus, you're able to balance that ratio of carbon to nitrogen. And that's the logic of the study, is that potentially mycorrhizal fungi can restore the quality of milkweed that may be in decline due to enrichment of atmospheric CO2. So briefly, here was the experimental design in our study system. You had milkweed, and the soil was inoculated with mycorrhizal fungi, and then you had 2 different CO2 levels. So the 6 species of milkweed included Asclepias incarnata, Selavantii, Syriaca, verticillata, Viridis, as well as tuberosa. The 2 CO2 conditions included ambient levels as well as 3.5 times ambient level. The reason I chose 3.5 times ambient level because that was a previous level that was found in the fossil record. The presence and absence of mycorrhizal fungi, so a mycorrhizal community where the inoculum was introduced into the soil, as well as sterile inoculum. And this was replicated, there was 8 replicates for a total of 192 plants. And the parameters that was measured included biomass, as well as root morphology. To measure root morphology, I'm going to introduce you to ways we classify roots. So down here, where you see these ones, these are considered first order roots. And these are the branches that are most distal from the main axis from the main root axis. So these are first order roots. First order roots are the roots that are typically colonized by mycorrhizal fungi, as well as the roots that's able to take up the soil solution as well as nutrients. Okay, and then when you see when you count backwards from 1, that's the first order root, then you go back to 2, the second order root, and you get back to this main axis root, which is number 3 here. That's the highest order root. The function of the highest order root is mainly conductance as well as storage. So that's where you have that's where the carbohydrates are more likely to be stored. If you're familiar with a carrot root system, that's where you would have a tuber forming. And so to measure root morphology, I use the difference in the diameter of the transport root versus that of the absorptive root. And divided by transport roots, I gave us a relative difference and multiplied that by 100. Okay, time for the results. Briefly, I'm going to show you a qualitative result here. And for this qualitative result, the 4 species on the left side are your taproot species, meaning they form, they have a root system where there's a dominant root with like a main axis. And then the 2 species on your, in Zoom world, I'm guessing it's on your right as well, on your left. Just the 2 last species on the screen, I'm not sure how you guys think. Those 2 species have a fibrous root system, meaning they don't have a dominant root. And then on the top panel where it says on your y-axis, we see plus, that's going to be the presence of mycorrhizal fungi. So that's going to be the effect of mycorrhizae on the roots. And then you're going to see the absence of mycorrhizal fungi. That's the control, if you will. Okay, so qualitatively, what did I observe? As you could see that the roots on the top panel, meaning the presence of mycorrhizal fungi, increase the root diameter all around, as well as there was more branching. And that's what we saw all across, regardless of root system, whether or not you use a tap root system or a fibrous root system. If you're a milkweed, your roots were more pronounced with mycorrhizal fungi. In the bottom panel, you see the absence of mycorrhizal fungi led to roots with less vigor. And so that's the effect of mycorrhizal fungi, as you can see. And then you can see that the fibrous roots on the two species, Incarnata and verticillata, on the far right side of the screen, those do not have a primary dominant root, such as the four on the left. So these are the fibrous root systems. Okay, quantitatively, what did we see? With regard to the root system, there was no effect of atmospheric CO2 on the root system. In terms of relative transport of root, that's the difference, how different the transport roots, which are involved in storage, how different they were from the absorptive roots that are involved in soil solution uptake, nutrient uptake, and those are the roots that are colonized by mycorrhizal fungi. You see that the presence of mycorrhizal fungi led to increasing difference between the transport root as well as the absorptive. And when you look at this, when you look at the effect of CO2 and mycorrhizae on relative transport route, there's an interaction as well as significance for both CO2 and mycorrhizae. So for those of you looking at verticillata here, you can see that there's an interaction such that when there's enriched atmospheric CO2, the effect of mycorrhizal fungi, which is the yellow, it declines. Whereas when there's ambient CO2, the effect of mycorrhizal fungi is stronger. Okay, now for the top left panel, you see we're looking at biomass now. Enriched atmospheric CO2 depressed the biomass of the milkweed species, while mycorrhizal fungi had the opposite effect and improved biomass. In terms of the different species, how these two factors of mycorrhizae and CO2, how that impacted the biomass of these species. One way to look at it is that these species here, tuberosa, Sylvantii, and Incarnata, that's that top. Those are the three species that make up this top angle, if you will. You see that for the right yellow box plot, you see that when you move from ambient, let's see if I use the pointer here. We go from ambient to enriched with tuberosa. You see that there's a drop off in the effect of mycorrhizal fungi. That is to say that the yellow box plot declines when you go from ambient to enriched. And there's a similar pattern for E, which is Incarnata up here. And there's a similar pattern for Sylvantii. These three species have something in common. Not only are the effects of mycorrhizal fungi being depressed under enriched atmospheric CO2, but these three species are also less weedier. They're more case strategists. So we're seeing that the less weedier species, the case strategists, are being more negatively impacted than the other three species by enriched atmospheric CO2. So that means that non-weedy species, the mutualism that they're gaining from mycorrhizal fungi is being lessened under enriched atmospheric CO2. And there was a correlation between relative transport route and biomass, and this was significant. Also, as it relates to relative transport route and biomass, we see that these are all the different plants used in the study. You see that there's a difference. There's less overlap between the presence of mycorrhizae and that of the absence of mycorrhizae. There's a lot of overlap between CO2 when looking at this relationship between biomass and relative transport route. And this right here, looking at the different species, might be confusing to look at all 6 species at once. But when you assort them by root system, you see that they're going in different directions. One is going in a steep positive correlation direction, such as a 45-degree angle, whereas the other one going close to vertical. Okay, because the nature of this study was looking at how CO2 can impact the milkweed species, we did a phylogenetic analysis where we looked at the relationship of the milkweed species by using a CO2 fixation gene called RBCL, and this is the chloroplast gene. And interestingly, the 2 fibrous root species are sorted together. So in other words, this is the first time I saw it presented like this, that the species are actually could be observed as being related according to their root system, where the 2 species that are most closely related actually have fibrous root systems. And these other 4 species have taproot systems. And this phylogeny is actually consistent with previous phylogenies that looked at milkweed species. When this paper was published, the author was actually interested in the rhizal morphology. That's why you're seeing these gray squares versus circles. So clonal, which is the gray squares, meant that they wanted to know if the species are sorted by a rhizome presence, a large rhizome. And they found that the large rhizome presence did not explain the phylogeny. As you can see that with some of the red species that are used in this studies are the ones with the large rhizomes, whereas the circles are with not a large rhizome. And you see that the rhizome presence does not explain this trait. But interestingly, due to the nature of our research question, we was able to observe that root systems are actually explaining the phylogeny, at least with the species that we observed. Okay, take home points for this portion of the talk. Mycorrhizae increases biomass while enriched CO2 decreases it. Interactions with CO2 and mycorrhizae was observed for both root and morphology, root morphology as well as biomass. We saw a relationship with the CO2 fixation gene and phylogeny, and the effect of mycorrhizae was most pronounced in taproot systems, and there was a strong positive correlation between relative transport route and biomass. Okay, so back to the overarching theme of my research agenda. Do microbes mitigate? Sort of. We saw that CO2 depressed biomass while mycorrhizae improved it, but we say sort of because there was an interaction, and it may be due to the milkweed life history. As you saw that when you were a case strategist, a non-weedy species, your interaction with mycorrhizae and CO2 was different when you were a weedy. Not saying that you guys could turn into a milkweed, but I'm just saying you, and yes, it's easier to talk about that. Okay, now for the second part of the talk, do microbes respond? And sort of like first responders, I would expect they'll be able to mitigate the flames of environmental change. And so for the second portion of this talk, I'm going to turn to trees that associate with ectomycorrhizal fungi versus arbuscular mycorrhizal fungi. And I'm interested in how the decomposers respond. Okay, so this is a figure from a 2019 paper published by the Rich Phillips group at Indiana University. And what they're looking at these different ecoregions. They classify such as 9 ecoregions, and they're interested in the change from forest stands that are dominated by ectomycorrhizal fungi versus forest stands that are dominated by arbuscular mycorrhizal fungi. And so what you see in a lot of forest systems is an increasing shift from ectomycorrhizal dominated trees to arbuscular mycorrhizal dominated trees. Some of the explanations from this that may explain this is increase in nitrogen deposition, as well as fire suppression, such as on the East Coast, white oak, which is an ectomycorrhizal associated tree, it's not regenerated because it's usually dependent on fire to clear the understory a bit and allow it to regenerate. So when you look in the understory, you see a lot of sugar maples, red maples, arbuscular mycorrhizae. So you're getting a shift in from EM dominance to AM dominance. And this is due to, in some cases, a lack of EM trees regenerating. You don't see as much saplings, and there's nitrogen deposition, as well as enriched atmospheric CO2 can lead to changes in precipitation, and then you have species that differ in their mechanisms of acquiring moisture from the soil and things like that. Okay, so, and the reason this is important for my study system is there's also another difference between arbuscular mycorrhizal and ectomycorrhizal associations. Ectomycorrhizae are descendants of brown rot fungi. Brown rot fungi are able to degrade cellulose, and therefore they contribute to wood decomposition, where you have the brown rot fungi degrading the cellulose, and you have white rot fungi degrading the lignin. Because ectomycorrhizal fungi are descendants of brown rot fungi, that means they have lignocellulitic capabilities. They could span this biotroph saprotroph continuum where they're biotrophs, they're obligates, they're mutualists, but they could also modify soil organic matter and contribute as decomposers. Interestingly, what this group also saw that there was a decrease in C to N ratios as they're with relation to AM tree dominance. So this brings me to this question: will change in predominant mycorrhizal type impact recalcitrant litter decomposition? And to address recalcitrant litter decomposition, I'll be looking at wood. And I'll be looking at wood decomposition with relation to the two different mycorrhizal types and how wood is degraded. And also I'll take into account that the ectomycorrhizal and arbuscular mycorrhizal trees, they also have different canopies, meaning that they contribute different types of litter to the soil environment, such as broadleaves versus needles. And also there's this brown food web effects, such as the role of the macroinvertebrates. And also, because I'm just such a rooty type of guy, root morphology might be important as well. So here was the 13 species that were used in the study. So altogether, there was about 28 observations for arbuscular mycorrhizal trees. So this is wood that's decomposing within the root zone under these trees. Another way of putting it, wood is decomposing beneath the canopy of Asus saccharum, Prunus serratna, and they're decomposing slightly beneath the soil surface, and they're in contact with the roots and the different mycorrhizal associations. So we're comparing how the root zone mycorrhizal associations of arbuscular mycorrhizal fungi, which is all the EM species in the top panel, versus that of the ectomycorrhizal species in the bottom panel. And also at this site in Pennsylvania, we previously measured the root diameter. So fine root species, which I'll be referring to later in the talk, are species with an average of the first order root or the first three root orders being less than 0.33 millimeters. And coarse root species are species that have an average root diameter of the first three order roots that's greater than 0.33 millimeters. Okay, so this is the effect of how we excluded the macroinvertebrates. And here's a mesh that's about 0.22 millimeters. and it's able to exclude the larger macroinvertebrates. And this wood substrate, the wood substrate next to it, without the mesh, is able to be, it's not, macroinvertebrates are not being excluded or introduced. And this cylindrical core with all these windows, in previous studies, I found that it's permissible to salamanders, to all sorts of large critters in my previous studies. So you get spiders laying eggs in there, and you get salamanders jumping out at you, and it was an interesting dissertation when I worked with this in previous studies. So what's being manipulated here is the exclusion of macroinvertebrates, and these cylindrical cores are filled in with the rhizosphere soil from the canopy tree, which is underneath it. And this happens for about 13 months. So although we create a disturbance, everything comes back to sort of a natural condition after 13 months. The hyphae and fungi are able to grow in here and decompose the wood as expected. Okay, as for the results, here was an interesting association. So on your x-axis, you have root diameter. You have these fine root species on your left. The fine root species are the ones that I'm identifying as less than 32 millimeters in their first 3 root orders. And then you have your coarse roots. And as you go from fine root to coarse root, interestingly, we saw an increase in wood in relative wood decomposition. So this is the change in mass. We weighed the initial mass of the wood, came back 13 months later, and we're measuring what's left. And this is what we found. So this suggests that as you increase root diameter, there's an increase in decomposition. Now, what might be explaining this is that if you're a fine root species, you're probably able to acquire more soil moisture. And as you reduce soil moisture, therefore, you reduce an important component of decomposition, which is moisture. So that may explain why the fine root species are slowing down decomposition. They're making the soil more dry. And when we group this according to coarse root species versus the fine root species, decomposition, as you expect from the previous figure, was greatest when the wood was decomposing beneath in the soil next to coarse roots versus fine roots. We're looking at all combinations of mycorrhizal, AM versus EM, and just running the model. There was no difference between AM versus EM. And also, there was a significant effect of macroinvertebrate exclusion. So this says that when we excluded the macroinvertebrates, there was more decomposition. This may suggest that the macroinvertebrates that were abundant at this site may have been disrupting the decomposers, whether or not it's their mycelial networks, their hyphal networks, or whether or not they're actually grazing on them directly. Because I didn't quantify the abundance, I can't say which mechanism they're using to affect what's going on, but we see that when you exclude the macroinvertebrates, there's actually more wood decomposition. And this is how it looked like in our ANOVA analysis, and we didn't detect any interactions. The macroinvertebrates and root morphology was significant, while mycorrhizal type and foliar litter did not affect decomposer response. So the working model of this experiment, I wanted to know that mycorrhizal type, the foliar litter input from the canopy tree, the tree that's directly above the soil, that was impacting wood decomposition, as well as the macroinvertebrates and root morphology. What I found was that there's a significant effect of macroinvertebrate exclusion, as well as a significant effect between root morphology. That's the fine root species versus the coarse root species. So take home point, macroinvertebrate exclusion significantly increased wood decomposition by 5.9%, while mycorrhizal type did not affect wood decomposition, nor did foliar litter input. A marginally significant effect was observed when you just looked at the change in root diameter. That was the first plot I showed you with the Pearson correlation. But when you group it by two categories of coarse roots versus fine root species, we saw that there was a significant difference in decomposition. With decomposition was increased by about 8%. Okay. To make sense of the last portion of my research agenda, so do microbes respond? And I'm talking about the specialist decomposers decomposing wood. We saw that they responded to root morphology, the coarse roots, as well as the exclusion of macroinvertebrates. And so with that being said, I'd like to acknowledge my funding source, the NSF Postdoctoral Research Fellowship in Biology, which I started on. And I'd like to acknowledge NIH Aracta, where I'm a fellow with Dr. Roberto de Guzman. And also I'd like to acknowledge my co-authors for the two studies I mentioned, Jim Bever, Dave Eisenstadt, Terry Bell, and Marianne Bruns. And with that being said, I'd be delighted to take any questions. Thank you.
Okay, I will try and man the thing here. So I'll just keep your slideshow up in case you want to use stuff to navigate that. And does anybody in the audience here have any questions, or out in Zoom land, just do the hand raising thing and we'll. Kelly, do you have a question?
I do. I was wondering if you thought about looking at those carbohydrates and if you had any guess on how they might be affected in your experiment.
Can you say how if he had looked at what, Kelly?
The cardinalides and the milkweeds on the first experiment.
Okay. So whether he had looked at the cardinalides, for those of you that are out here, I know my speakers aren't great. So if he looked at those cardinalides that the monarchs.
Yes. So Kelly, as to whether or not I looked at the cardinalides, that's actually the next part of the study that's underway. We decided that it would be best to look at the cardinalides at the same time as looking at the monarch larvae that are actually participating in herbivory. So we want to make sure that because the monarchs are dependent on the milkweed and cardinalides, we thought it would be an interesting study if we look at the cardinalides and the monarch larvae grazing at the same time. So we didn't look at it in this study, but that's actually what's happening next. Thank you.